Right. So I'm very excited to introduce Charlie Thrift. He's a PhD student at UC Santa Barbara in Dr. Katja Seltmann's lab, and he's co-advised by Hilary Young. And he said that he first became interested in bees when his mother was very afraid of bees. So as an overcorrection, he got really excited about them, going to a camp at the Santa Barbara Natural History Museum while he was in high school. And now in his PhD, one of probably the many projects he's working on, he's looking at extinction risk factors for native bees, including their diet. And that's what he'll be talking about us tonight. So with that, go ahead and take it away, Charlie.
All right, thank you. Well, thank you, Jess, for that great introduction, and thanks everyone for being here on a Tuesday night. Very excited to share some research with you all on some concentrated vulnerabilities in bees. Diet specialists have smaller geographic ranges. And this work was co-authored by some really incredible scientists, including Thomas Wood, Anne Bowie, Hilary Young, and of course, Katja Seltmann, who is my main mentor, advisor, expert on all things bees, research, academia, and I'm so grateful to be a student working in her lab. All right, so as Jess said, my journey into bees starts during high school, really, and I am so sorry to say to this Zoom room full of people involved honeybees at first. So in my high school, we actually had an apiary, and so I got to get some experience doing beekeeping and all that entails, and really figured out that this was so cool. You know, these insects are so fascinating, and all of this good stuff. And during that same time, I was also a teen participant in a teen science program through the Santa Barbara Museum of Natural History called Quasars to Sea Stars. And through that 4 year program, I sort of got to figure out that there's this whole thing that exists called ecology that looks something like this, where you can actually go work outside in some beautiful place and collect data and learn more about the world around us. And so I got totally hooked on that as a concept. Um, and then also started to gain this really vast appreciation for natural history collections and for museums. And you know, this discovery that we have millions of these little pieces of data of objects of biological, paleontological, geological evidence of a thing occurring in a place at a time. And combined, we can answer and ask so many really great questions using these specimens. All we have to do is take care of them and do things like digitize them or take pictures of them. And sort of this knowledge that as our technology advances, as our background knowledge and all kinds of other fields advances, the same specimens can be used over and over and over again to answer totally new and pressing and important questions. And so I gained this really great appreciation for museums during that time. So that takes me to my undergrad at UC Santa Barbara, where I was working in the Cheadle Center for Biodiversity and Ecological Restoration with Dr. Seltmann. And it was during this time that I got to appreciate and understand and experience that there are so many other bee species out there besides that honeybee. So we're no more honeybees for the rest of the talk, don't worry. Only the native ones. And so during this time, I was involved in bee curation and got to do some collection and some actual field work, which was super exciting on Santa Cruz Island, of all places, as well as some other places around campus. Um, and so I got more and more hooked on bees and ecology and sort of asking and answering questions in this space. My sort of summer 2020 lockdown project years later became this publication with a lot of help from Dr. Madeline Oswald, who was a postdoc at the time in the lab and is now a professor at the Queen Mary College in London, where we were really just honing in on this one species of Halictus bee, both on Santa Cruz Island and on the mainland, looking at their wings and looking for some population level differences there. So it was more of a taxonomy systematics type of project. So when I came back to UC Santa Barbara, spoiler for grad school, um, I think my interest in bees is really strong. I really like bees. I could have very happily been an entomologist, focusing on these over 20,000 species of bees that exist and continuing with some of this like taxonomy, systematics types of work. But we're living in this time of extreme biodiversity loss. And so we are losing species, and a lot of species are threatened and in decline. And because of this, my research is really motivated by this idea of informing our bee conservation efforts. So some of my graduate research looks like getting up close and personal with bees while they are alive in the fields, as well as back in the lab, working with often undergraduate students, taking them into the fields to places like Santa Cruz Islands or the Eastern Sierra Nevada, as well as all the research I'm going to talk about today, which I don't have a slide for, or I don't have a picture for because I have all the rest of the slide deck for that. But really interested in figuring out how we can better conserve, better save all of these species. And so the problem, or one of the problems here, is that we know wild bees are at risk. And by the way, we know that wild bees are incredibly, incredibly important. I'm not going to spend time tonight trying to convince all of you of that. We know of their importance. But we know that they're at risk. We know that species are declining, species are being lost. But the problem is we don't know really which species these ones are. And we need to know which ones are declining, of course, so that we can focus our conservation efforts on those species so we can triage our conservation. So what do I mean by we don't know which species are at risk? Well, bees as a whole are really under-assessed for extinction risk. So if you're familiar with something like our IUCN Red List from the International Union for the Conservation of Nature, they offer this great like scoring card matrix type of framework where we can place a species somewhere on this continuum from least concern, so it's doing fine, to near threatened, threatened, which includes things like being endangered, vulnerable, to all the way to extinct in the wild or extinct as a whole. So for bees, of our over 20,000 different species we have, most of them are actually way out here and either are data deficient or are not assessed at all categories. In fact, 97% of bee species belong to one of these two categories. So why do we not just go ahead and assess the rest of the bees? Well, the reason that they're in here, I like to say that they are small and they move fast, which I think is true for most bees. Um, but also partly because our IUCN Red List criteria doesn't really work that well for bees. So here's just a snapshot of what their criteria look like. I know we're on a variety of screen sizes. It's a lot of small text, so I'm not asking you to read it all. But they use criteria like population size reductions or geographic range size or actual knowledge of exact numbers of populations. And knowing these types of things for bees in particular is really tricky. So knowing the population size of any insect is quite difficult. They fluctuate naturally year to year. There's some boom and bust years. Figuring out how we can actually guess at what these populations sizes look like. Be great if someone can come up with some new methods for us to do that. But we're not going to just go count them one by one. And the other point I want to emphasize here is that this sort of scorecard, if you will, is the same exact scoring system for bees as for any other animal. So if we're trying to assess a polar bear versus if we're trying to assess one of our mining bees, we're going to use the same criteria including things like population size reduction, exact number of mature individuals in the wild is one of our metrics on here, which really have vastly different meanings and especially magnitudes in terms of insects versus some non-insects. So some awesome folks are leading great efforts to use these criteria to assess all the bees and try to get things as much as we can out of the data deficient category or out of the not at all assessed categories so that we can better understand how they're doing. But where I see my research fitting in here and what I'm really motivated to do is to sort of go sidestep some of these criteria and think about other ways that we can tell if a species is vulnerable or doing fine. And so I'm interested in using some other traits. So things that aren't necessarily in here, but might be really informative, especially for bees. So as we know, bees are diverse. One of the reasons that so many of us love bees is because there's all these, I mean, we saw our photo share earlier, so many different awesome shapes and sizes and colors of bees. So bees can be big or small at the species level, and also within a species, there can be variation. They can come in different colors. Hairiness is a really cool trait. Some bees are super, super hairy and fuzzy. Some bees not at all. And nesting. So a lot of bees are nesting in holes in the ground that they dig themselves, but some bees will nest in wood or in stems, or there'll be sort of these renters where they go and use someone else's, like maybe an abandoned rodent burrow or something like that, these cavities. Bees have different social socialities by species. So some bees, like our honeybees and our bumblebees, are going to be social, where they have a queen and some female workers, and there's this division of labor. But the vast majority of our bee species are solitary. So each female will make her own nest, provision her own nest, and the males are largely deadbeats. So we can also think about bees being diverse in terms of their flight windows. So maybe this is a little bit more in the weeds, but most bees are only active for a short time out of the year, maybe a few weeks, where the adult is actually flying around. Um, but some bees will be active for a much longer period of time, so months, several months at a time. And other traits would be things like thermal tolerance. So how warm or how cold can a given species get? And then some other things like range size and diet breadth. Foreshadowing, we're gonna get into those as well. But my point here is that beyond just those initial IUCN criteria, there's a lot of different ways that we can sort and classify our bees in a lot of ways that are going to be really meaningful from a conservation perspective. So I want to argue that we can assess bees with a trait-based approach. So instead of only using some of these IUCN criteria like population size, we can use some of these other things. Today, I want to talk about diet breadth. So getting into the trait of bee diet breadth, and Crystal gave a little intro to this earlier, talking about the 7-Eleven Slurpee example, which is perfect. But our adult bees are flying around, they're going to flowers, they're eating nectar for themselves usually, and then the females will collect pollen from the flowers so they can take that pollen back to their nests. And then they can provision their nests with that really incredible protein food source for their baby bees. So then when those eggs hatch and the larvae come out, they can eat that pollen. Some bees are not picky, so they will go to any old flower. They'll go to the Trader Joe's, the Albertsons, the Vons. They'll go eat a 7-Eleven hot dog. They're fine with it. Other bees are a little bit more picky, and so they're only going organic. They're only at the farmer's market, something like that. And so we'll often divide these types of bees up by being either a specialist. So we're thinking about bees that are visiting flowers within one plant family, one botanical family. Or generalists would be species that are visiting and collecting pollen from flowers from more than one botanical family. And of course, in biology, anytime we have sort of this either or, there's always someone else doing something totally different. And so there are our kleptoparasitic or cuckoo bees, which instead of collecting their own pollen for their young, they just go ahead and steal from the other bees. So they'll lay their eggs in someone else's nest where pollen has already been collected for them. So one species will be a parasite on the other species of bee. And this is about 13% of all bee species that have this lifestyle. And so for the rest of the talk, we're going to focus really on the bees that are actually collecting their own food. So these categories can be really informative especially when we have those data available. But I'm really interested in thinking about this trait as a little bit more of a continuum rather than an either or. So just to give an example of what I mean by this, when we think about a bee like our diadasia bee on the left, it's often thought of as a specialist, particularly on things like mallow um, and plants in that family, versus someone like our nice Mellistodes over here. This species is more of a generalist, and so it can visit multiple families. And that would be our categorical diet, so a specialist or generalist. But there's other ways that we can think about the diet breadth, including more of a numerical approach or a phylogenetic even approach. And so if we actually count the number of plants, we could do species, we could do genera, we could do plant families that bee is actually visiting and collecting pollen from. We can sort of compare within these categories as well as instead of just saying specialist or generalist. So we can move towards this framework where we can say a specialist that is visiting a single plant species. It can be super, super specialized. A different specialist that visits a dozen different plant species within a family would be maybe a little bit less so, but still in that sort of specialist side of our number line. And then a generalist, you can picture a bee that's visiting two plant species only, but they're from different plant families as being sort of in the middle compared to a generalist that's going to dozens of plant species from dozens of different plant families. And so one of the reasons why we don't see more of our understanding of diet breadth in sort of this continuum number line type of setting is because we don't often have the data to actually say how many host plants does this species have, or what is, for the phylogenetic side, what is the evolutionary diversity of all the host plants that bee is visiting. And so, let's see, using some bees do have preferences, absolutely. So using some really great pollen association data sets, we can move from this categorical to more of this numerical or phylogenetic approach. And so for the research I want to share with you today, thinking again through this trait-based conservation focus for bees, this really simple, I think, idea of do these conservation relevant traits correlate within species? And so if they do, if there's this positive correlation, so positive meaning as one increases, the other increases, not necessarily good correlation, that could concentrate risk in bee species, which would also allow us maybe to prioritize those species. So things that have for example, a risky trait value in one trait, as well as a risky trait value in a different trait, that make that species particularly vulnerable. As opposed to if there's a negative correlation, that might distribute some of this risk across bee species. And so, these positive correlations are sort of in line with niche breadth theory, which was proposed sort of in great detail in 1984, which would basically say, yeah, if something has a broad value here, it'll probably have a broad value somewhere else. But this wasn't known in bees, particularly because our understanding of bee diet breadth is quite limited. So we only know, if a bee is a specialist or generalist for a small percentage, maybe about 5% of our bee species that exist out there, of our pollen collecting bees. So my research that I want to share is concentrated vulnerabilities in bees. Diet specialists have smaller geographic ranges. So I'm giving everything away in the title. So sorry, there's no suspense to be built here. But this was just published in Insect Conservation and Diversity. And so, motivations for getting at this question in bees, we're starting to sort of lay out. And I want to emphasize again that these different traits can inform extinction risk and can inform the conservation relevance of these species. So, for example, range size independently will inform extinction risk in a species. And this should make sense, where small range size species are going to be more extinction prone than a large range species. So if you picture, a bee that is existing all the way across North America, so that species is all over the place. It's less likely that one single event, so that could be something anthropogenic, that could be something natural, maybe a volcano erupts somewhere. It's less likely that that one event is going to affect all the individuals of that species at that time. Compare that species, it's all across North America, to a bee species that has made its home on a nice little mountain called Mount St. Helens. Maybe that bee is doing totally fine, but then maybe in 1980 there's an event that might affect all of the individuals of that species. Um, and also geographic range size, again, this is used directly in IUCN assessment criteria. So this makes sense where smaller range species are potentially going to be more vulnerable. So another trait, diet breadth, also independently of range size informs extinction risk. So specialists are going to be more vulnerable to anthropogenic change than generalists. And this is true across the board. So these are both review papers that are really, really broad, really excellent, but also proven individually within bees. And I want to make a point here that specialization isn't like a bad strategy or something like that. Often in evolutionary history, specialization is an excellent strategy. If you become really good at the coupons at the Albertsons, you're going to be getting some great deals, some great groceries. But when we have something like anthropogenic change really accelerating some of the conditions that are driving, who's doing well, depending on which grocery stores you're going to, if you're not able to pivot to something else, then you'll be more vulnerable. So mechanistically, being a specialist, not a bad thing. But if you're depending on certain plants and then those plants are going extinct or they're changing their phenologies for some reason or the other, then you might be left hanging if you can't switch to something else the way that a generalist would. So then my question here would be, do both of these traits that independently inform extinction risk, do they correlate? And that would be, are they correlating in line with niche breadth theory? So we can look at some other species to sort of get an idea of how they are doing with these trends. So butterflies and moths have a positive correlation between range size and diet breadth, such that things that are more generalized, so eating lots of different foods, tend to have larger geographic ranges. The same is true for rodents and shrews, as well as primates. However, frogs have sort of the opposite trend. They have a negative relationship. So frogs that have broader diet breadths tend to have smaller geographic ranges. And then no trend is really found in bats. And then bird studies have shown some conflicting results. So nothing super conclusive across the animal kingdom, but of course some good examples of positive associations. So we want to look at how this relates in bees. And again, the reason why we haven't been able to ask this yet, or before now, is because we don't have, or we didn't have this really broad understanding of diet breadth in bees, such that we could compare it with range size. And so to ask this question, step one, we just need to do assign diet breadth to a whole bunch of bees. So in 2023, Thomas Wood published this really great paper, which is our most comprehensive to date understanding of diet breadth in bees. So this was over 860 different species. And the way that he did this research was going to natural history collections, going to museums, taking their bee specimens and physically scraping pollen off of those bees. So in some cases, these bees were super old. They had just been a dead bug on a pin for a very long time. But he was able to scrape some pollen off of them or pull it out of their mouth for some of our things like our Hylaeus bees that like to hold pollen inside instead of on hair. And then looked at that pollen under the microscope and identified those pollen grains to their botanical genera, families, sometimes even species. So this is a really, really cool data set, really cool way to use natural history collections, of course. And so I saw this paper and I emailed Thomas would. It's just a cold email. And I said, this paper looks cool. I used some of the data that you published in this table, and I made this preliminary graph. Do you have any more data? And he very quickly replied and sent me this some other great data that didn't make it into the paper. And so that was a great great example of a cold email working out. So taking all of this impressive data and putting it into sort of a table that looks a little bit like this, where we can have our individual bee species. And for each species, we can assign it some of these diet breadth values. So a categorical diet, like specialist or generalist, as well as this numerical diet, actually counting for example, here, the plant families that bee was collecting pollen from. And you'll notice there's some decimals in here, like 6.482625. That species wasn't collecting from that exact number of plant families, of course, but that's just there to remind me to tell you that we did a rarefaction procedure with those data so that we're not confounding rarity in the data set with specialization. So if we only had a few individuals of that species, we do this correction so that we can say, a few individuals from one species compared with a whole bunch of individuals from another species. You might expect one of them to have just more diverse plants that they were collecting pollen from because there's more opportunity to in that data set. So that was rarefied. Great. Next, we need to assign range size to a whole bunch of bees from the same bee species. And so to do this, we use this really great validated occurrence data set published by James Dorey and co-authors in 2023. And we were calculating the extent of occurrence, or the EOO, for each species. And the extent of occurrence is a proxy for range size, basically where if we plot all the points of that species on a map, and I should say here, occurrence data is really just data on a given species existing at a time in a place. And so if we say that a bee species was existing in all of these different points, these black points on this map in this figure, then we could connect the dots, and that shaded area in the middle can be our range size or our extent of occurrence for this species. Now, you may be thinking, this doesn't look like a perfect example of range size in a species, and that is great. Please, by all means, keep thinking it. And then when you have a better idea, let me know, and we can go from there. But the extent of occurrence is directly used in some IUCN assessment criteria, which is one of my motivations for using it. And it does make a little bit better sense than something like the area of occupancy with the data set that I was using. So by all means, we could do other creative things like build these species distribution models or consider habitats and discontinuities before we just connect the dots. But for this research, we're using extent of occurrence. So when I say range size, from here on out, I'm talking about this sort of purple blob idea. All right, so then we just need to combine our diet and our range data. And so what we ended up with was 633 bee species from 6 different bee families. We have all the families except for Stenotrididae, which is a very small, only 21 species, only in Australia. Sorry to anyone from Australia listening, Elaine, if you're here. Diet breadths. So we have diet breadths for these species, categorical diet, so that's that specialist versus generalist, and then numeric, phylogenetic, and then we also have range size as the extent of occurrence. So as a reminder, what we're looking for, does it go up, down, or dash line with no trend for our bees? So combining diet and range size. We were able to assess this relationship with all 3 of these diet categories, or diets classifications, so the categorical, the numeric, and the phylogenetic. We find the same trend with all 3. So I'm just going to show you the numeric so that we don't have to see repeats of the same types of graphs. So I'm going to show one graph here where we have on our x-axis diet breadth ranging from specialized to generalized. And then on our y-axis, we have range size ranging from small to large. And so a point is going to be a bee species. So it's a bee species that has some value of diet, some value of range. And we're going to put all the points on the map, and we're hypothesizing that we'll see this positive trend, sort of in line with our butterflies and moths and some of those other creatures, as well as niche breadth theory. All right, so now to show you what the real graph looks like with 633 dots for our 633 species, it looks like this. And so, yes, we find that there is this positive correlation between range size and diet breadth in bees. And if you aren't super impressed with the slope of this line, if you think it looks a little shallow, I want you to consider, would you be comfortable getting on roller skates at the top of this, backwards, facing the other way, and going down? If the answer is no, then I hope you'll agree that this is a positive association. But we can zoom out a little bit, see the whole figure, which divides this into panels. So each color represents a different bee family in the data set. And with the models that we were building here, we're able to argue that this positive or increasing trend between range size and diet breadth in bees holds up among these 6 different families as well. And so some stats as well. So if we look at like our Spearman's non-parametric test, we get this moderately strong relationship between these 2 variables. And then the model that we're building here, which is this generalized linear model with the gamma error distribution and a log link. So I also want to point out real briefly that these axes on this graph here are not transformed in any way. But if you most of what's happening in this graph is sort of in this bottom left corner. So if we were to zoom in on that, or if you were to log transform both of the axes here, we'd see more of a clear positive trend. All right, so our range and diet positively correlated in bees, but we're finding similar results just by the way with our phylogenetic diet. So that's taking into account the evolutionary history and evolutionary relatedness of the host plants. So if you are a bee that's visiting 2 plants that are closely related, that would be a more specialized score than if you're visiting 2 plants, so the same numeric score, but they're very distantly related. So if you're going to the Albertsons and the Vaughns, they're kind of in the same tree, but if you're going to the Albertsons and the Trader Joe's, it's a little bit more diversity there. And we also looked at this with the PGLF regression, or phylogenetic generalized least squares. And so if when looking at those points, you were getting worried about the fact that, hey, those aren't really independent. Bee species are some are closely related and some are distantly related. That's what that regression sort of accounts for and controls for. And we find, again, similar results where our bees with a broader diet are tending to have a broader range size as well. All right. So I think this makes sense for a lot of different reasons why range and diet might correlate within bees. So generalists like these beautiful Ogochlorella sweat bees are able to visit diverse floral resources across habitats, across phenological periods. They're not really constrained by a given plant species, so they're able to have larger species level ranges. Compared with specialists like this nice cactus bee here, they really are constrained by the spatial and temporal availability of their host plant. It's worth noting as well that some host plants are super widespread. And so some specialists maybe aren't as constrained as others. But if you are doing a road trip and you will only go to the Costco food court, you really need to plan that out in terms of location as well as in terms of hours and sort of dodging some holidays. So that's a little bit of an analogy for what can sort of constrain and prevent these specialists from getting a much larger range. So bringing this back to our conservation efforts, knowing that these vulnerabilities exist, knowing that these trends correlate, we can better target our conservation on some of these more vulnerable bees, even when we only know the value of one of these traits. So for example, and this is just an example, if we have a bee species that's a generalist with a large range, we might predict that it's going to be doing better than others. Maybe it's less vulnerable to some current anthropogenic change, to some current climatic conditions. compared with maybe a specialist. Even when we don't know the range size of that specialist, we might want to consider that to be more vulnerable than something that was a generalist, for example. And so just to share some limitations from our current diet and distribution data, this figure here is showing where the records for, particularly for range size, are coming from this data set. So we used that Dory et al. Data set, which is over 6 million records. So these are individual bees that exist somewhere and have been digitized so that we can know where those species come from. And so we can see based on these warmer, more yellow colors on this graph that the majority of our data, vast majority, is coming from Europe and coming from North America. And so it would be great if we could get more records from the rest of the world, especially global South, parts of Asia. If you look at this map, we should see bees everywhere except for Antarctica. So we know there are, of course, bees throughout Africa. We just don't have those data readily available, readily digitized yet. So working towards digitizing more museum data, as well as continuing to collect and amass biodiversity data, is going to be really important. Additionally, we have 633 bee species, which sounds like a lot until you remember that there's about 26,000 different species out there. So it would be great, of course, if we can get broader sampling, both across the map as well as throughout bee taxonomy, as well as considering, incorporating some additional traits. So I talked a lot about, of course, diet and range size, but there's a whole suite of other traits that might be really informative from a conservation perspective as well. And then, of course, want to really emphasize more and more digitization of museum specimens is going to be super important, super impactful, so that we can really fill out the gaps on this map and just get a broader understanding of how this trend plays out from both the pollen side of things as well as the range size side of things. And there's also some really cool work that's recently coming out. Colleen Smith led a paper in 2025 on predictive modeling of bee diet breadths using occurrence data and interaction data sets. So with the global biotic information data set, if you're familiar with that. So more work and more, I think, really creative work in terms of broadening our understanding of diet as well as range size. It's going to be really important here. And I really want to drive home the indispensable role of our natural history collections in this research and just in our broader fight for the conservation of nature. You'll notice that these are not bees, these are grasshoppers. This is actually from a story that came out yesterday from the IUCN, from the International Union for the Conservation of Nature, where they really just put out this statement saying natural history collections are super, super, super important to everything that we do for conservation. So we need to continue to fund them and use them and digitize things and take care of these specimens so that we can continue to amass our biodiversity info, both for conservation as well as any other research. And so, what I really want to bring this back to is thinking about, so we find this relationship, this positive relationship between diet and range size, where, there's going to be this whole chunk of bee species that are potentially, doubly vulnerable to the effects of both small range and to diet specialization. And those are species that we can focus our conservation efforts on. Those are species that we can target. And when we remember that 97% of our bee species are either data deficient or not assessed at all, we do need to think creatively about how we're going to move forward and better understand these bees before they just go missing from our traps. So bee conservation, we need to know which species are important with time to intervene, with time to make efforts and make changes to preserve those species before it's too late. And so I really want us to think about some of these other creative ways besides just the IUCN criteria, besides just that little scorecard, where we can really get under an understanding of vulnerability in these bees. And so with that, I want to thank all of my awesome co-authors, especially Dr. Katja Seltmann, and some other folks and lab members for some great discussions on this project. And I think we have time for questions